So to back up a little, I mentioned the word phenotypes. Our understanding of phenotypes is only possible because of a NIH-sponsored group called the Severe Asthma Research Program, or the SARP. Some of you have heard of them. It's their description of severe asthmatics in a complete clinical, biological, and epidemiological manner that allowed us to appreciate that there are indeed subgroups of asthmatics. And these categories have been associated with different inflammatory mechanisms, different natural histories, and so you can imagine that maybe each subgroup will have its own therapy one day. So what's a phenotype, you might ask? Well, it's the observable characteristics of each individual that arise from the interaction between their genetic makeup and what they're exposed to in their environment. In other words, they have a trait that gets expressed based on their genes and their environments. So why are phenotypes such a hot topic? They're a hot topic because the SARP described 5 different phenotypes based on 628 different variables. That's impressive. These cluster phenotypes predicted the response to treatment. So we know that FEV1, which is what we use in the past, doesn't really predict response to treatment. It doesn't really predict outcomes either anymore. So this is actually a very pivotal way of looking at patients. So what do these clusters look like? Cluster 1 is a typical patient who has childhood onset asthma. They're atopic, they have mild disease. Cluster 2 sort of looks like the same. They preserve lung functions, but they use a little bit more medication. Cluster 3 patients are older. They don't have allergies. They're obese. They tend to do worse. They have a lot of exacerbations. And then the cluster 4 and 5 patients are the ones that present to us with severe obstruction. These patients tend to be on oral steroids. So now what about the novel inflammatory pathways that I mentioned? I'll discuss 2 of the unique pathways that our laboratory at the Intramural Severe Asthma Research Group at the NHLBI has discovered and targeted. Now there are other novel inflammatory pathways, but they haven't quite made it to the bedside yet. They're still bench. So I'm going to limit it in the interest of time to just these 2 pathways. The first pathway was the recognition of something called the peroxisome proliferator activated receptor gamma. And in English, this is actually a receptor that's throughout your body. It's been associated with a very large variety of diseases and sort of pops up in cancer fields and everywhere else too. The most important point is that it's already being targeted by a class of drugs called the thiazolidinediones, and these are drugs used to treat type 2 diabetes mellitus. And specifically, it's rosiglitazone and pioglitazone. These 2 drugs have been interestingly found to have in a lot of animal and now increasingly human studies to show that they target the inflammation we see in asthma. Also, their levels are increased in asthmatics. So this cartoon shows a multitude of ways that PPAR gamma agonists are felt to affect or impact the pathophysiology of asthma in experimental models. They decrease eosinophils and lymphocytes, as well as IgE and Th2-mediated interleukins. They decrease the airway changes, including smooth muscle thickening, collagen deposition, and basement membrane thickening, and it also decreases mucus. This is a reason why our group designed the first human study of pioglitazone in asthmatics. So we're currently enrolling for this clinical trial. We've limited inclusion to severe asthmatics because that's our mission, to develop novel treatments for severe asthmatics. It's a double-blind, randomized crossover trial, which means that every patient who gets enrolled will get the drug. It's 11 months long, and our primary endpoint is actually the Asthma Quality of Life Questionnaire. It's not the FEV1 anymore. The reason why is because we want to know how an asthmatic will function with their disease after taking this drug. So, if you or someone you know is interested, I'll put information up at the end of my talk, or just come and talk to me after the session. So the second exciting pathway is one that we were kind of surprised by ourselves. Apolipoproteins you've heard of in cardiovascular talks. It plays a crucial role in cholesterol metabolism, and these are proteins that are made in the liver, and they've been described to have functions outside of lipid metabolism as well. We, however, found that apolipoprotein E is involved in controlling goblet cell hyperplasia and airway hyperreactivity. This is one of the slides from the publication we had last year showing that the absence of ApoE was associated with significant increase in goblet cell hyperplasia compared with controls. It's also very significantly associated with increased airway hyperreactivity. So the question we asked was, if you give ApoE as a mimetic peptide to an animal model of asthma, will it decrease the asthmatic pathologic changes? You can see here that if you do administer inhaled ApoE mimetic, you can reduce goblet cell hyperplasia and decrease airway inflammation and hyperreactivity. So going back to the earlier cartoon that I showed you of what asthma looks like in a human, this makes it easier to breathe. Along with apolipoprotein E, we also found that there was an apolipoprotein A1 that had a role in inflammation. So this one protein actually is part of the HDL complex, or the high-density lipoprotein. This one actually has very interesting properties as well. It's an antioxidant, it's an antithrombotic, it's involved with endothelial function. But what interested us was it's also involved with the anti-inflammatory pathways that you see in asthma. Again, using an animal model, we showed that this prevents asthma development. It decreases airway inflammation, decreases goblet cell hyperplasia, and these are two crucial pathways in asthma. So how do they compare with one another? Well, in the lab, they seem to both decrease airway hyperreactivity as well as inflammation and goblet cell hyperplasia. Our hope is to create an inhaled version of either of these mimetics. And why inhaled? Well, when you give an inhaled therapy, not only are you giving it directly to where the injury is, in this case asthma, but you can get higher doses without the toxicity. So we're in the process actually of securing funding for developing these drugs. So what about alternate pathways? Smooth muscle's been targeted by bronchodilators, and it is felt to be pivotal in the management of asthma. The thing is, recently there was an article in the New England Journal of Medicine showing that bronchoconstriction, which means that the pipes squeeze or the smooth muscle contracts, that in and of itself seems to cause inflammation. So in the old days, we used to use a lot of short-acting and long-acting beta agonists. We've sort of fallen out of favor with that, but maybe we do need to go back to that, because if that causes inflammation and airway remodeling, then maybe we should be targeting that pathway as well independently. So immunomodulation, I'll just mention that omalizumab, we've been using it for a decade. It's effective. It's a classic example of tailored therapy. It really only works in atopic asthmatics, moderate to severe, and it is quite effective, although not in every case. Anti-TNF therapy, anti-IL-5 therapy, they don't work, they're not safe, they're not effective, but anti-IL-4 therapies are being created, and look for that in the future. So I didn't want to finish without talking about comorbidities. These are very important. I will just mention that you should definitely think about each one of these and treat them. And if you are not sure, send them to someone else, because it isn't just as simple as giving a PPI to someone who's got reflux or sending them for a sleep study if they've got sleep apnea. These need to be properly managed, and you've got, luckily, plenty of help in this area. So I hope I've convinced you that not all asthmatics are the same. I hope you don't go away from here thinking the same cookie-cutter therapy for each asthmatic is appropriate. What I do mostly hope is that you walk away from here knowing that there are some newer anti-inflammatory pathways that are coming, that are exciting, that we are going to start phenotyping patients and offering them treatment based on that. This is where I work. It's the intramural campus of the National Institutes of Health. This is a large red brick building where we do all our translational research and we see all of our patients. This is the Severe Asthma Research Group, and these are the people who are responsible for all the exciting work that I showed you today. And finally, I want to thank you, the community, because without your support and referrals, we would not be able to do this cutting-edge research. Without your referrals, we wouldn't have the samples or be able to populate our clinical trials, and without you, honestly, there would be no future of asthma therapy.